Hello guys, I am Shubham from Tutorials. This video is all about proteins. So before we start, subscribe to our channel and press the bell icon to get notified whenever new videos out. This is absolutely free from your side, but it will help us grow. When I say all about proteins, means the things that are very important for the exam that I will be showing you, and this will be a long video, but it is very useful because protein ये complex topic है, but we will make it simple as much as possible, and I will give all the important things for exam. Okay, so let's start. Basically amino acids हमने देखा है, you will find it in the biochemistry playlist. So amino acids are the building blocks for protein, right? जो peptide bond form करते हैं. Okay, peptide bond form करते हैं, right? So amino acids are linked together by peptide bond and then they form proteins. Okay, so proteins are basically what? Polypeptides, right? Polypeptide chains. तो अभी इस वीडियो में स्पेशली हम स्ट्रक्चर्स देखेंगे प्रोटीन का, राइट? प्रोटीन स्ट्रक्चर. So there are 4 types of structure: primary, secondary, tertiary and quaternary structure, right? A description of all covalent bonds, mainly peptide bond and disulfide bond, linking amino acid residues in a polypeptide chain is a primary sequence structure. Okay, primary structure means just chain of amino acids होता है जिसमें bonds होते हैं, peptide bonds and disulfide bonds. So the most important element of primary structure is what? Sequence of amino acid, right? Primary structure में sequence होता है. Then secondary structure refers to particularly stable arrangements of amino acid residues giving rise to recurring structural patterns, right? Secondary structures में हम arrangement देखते हैं, okay, stable arrangement, and that gives rise to a pattern, specific pattern. Tertiary structure describe all aspects of the three-dimensional folding. See, folding in 3D space, right, of a polypeptide. When a protein has 2 or more polypeptide subunits, the arrangement in space is referred to as quaternary structure. Okay, quaternary structure means different subunits are joined together, right, and that arrangement is called quaternary structure. So our exploration of protein will eventually include complex protein machines consisting of dozens to thousands of subunits. So there are many proteins, right? Variety of proteins exist. Okay. One more thing. See, this is also definition. Secondary structure is the local spatial arrangement of a polypeptide's backbone atoms without regard to conformation of its side chain, right? Then tertiary structure refers to three-dimensional structure. Okay, 3D structure कैसा है? Of an entire polypeptide including its side chain. Many proteins are composed of 2 or more polypeptide chains, loosely referred to as subunits. Right? मतलब proteins में सिर्फ एक chain नहीं होते. Right? There are more than 2 or more chains also present. So that we called subunits basically. And quaternary structure is what? Arrangement of those subunits as a whole, right? As a unit basically. A protein's quaternary structure refers to spatial arrangement of its subunits, right? Subunits का arrangement कैसा है in space, right? So these are the structures basically. This video is all about structure. Primary structure first, then secondary structure, then tertiary and then quaternary. So now, see, this is one important chart. यहाँ से क्वेश्चंस भी आए हैं बहुत. Okay, So these enzymes are basically proteases. Proteases को यूज करते हैं फॉर एनालिसिस ऑफ प्राइमरी स्ट्रक्चर. ओके, प्राइमरी स्ट्रक्चर समझने में, दैट इज सीक्वेंस ऑफ अमीनो एसिड. ओके, अमीनो एसिड सीक्वेंस समझता है. So these enzymes are used. Okay, and these enzymes are very important. Like trypsin, right? Trypsin का cleavage point क्या है? Lysine arginine. Okay, lysine arginine, and C stands for C terminus, right? Then chymotrypsin, you have to remember aromatic amino acids. Okay, aromatic amino acid. These are aromatic amino acids: phenylalanine, tryptophan, tyrosine. Right? I will give you इसका shortcut है wife. Okay, F stands for phenylalanine, Y stands for tyrosine, and W stands for tryptophan. Okay, again C terminus. These two are the important enzymes. Lysine arginine को हम KR, right? K is lysine, R is arginine. Trypsin and chymotrypsin are the most important ones. You have to remember. बाकी ये Staphylococcus aureus, इसका target है aspartate glutamate, C terminus. Then again aspartate and protease है from bacterium Pseudomonas. Again same aspartate glutamate but N terminus, right? Pepsin cleaves at leucine, phenylalanine, tryptophan, and tyrosine N terminus. Then endoproteinase, next enzyme, lyse C, that is bacterium Lysobacter enzymogens. तो इसमें lysine cleavage point है, lysine. Cyanogen bromide is also important. Cyanogen bromide cleaves at methionine, C terminus. Okay, so this is also important enzyme, cyanogen bromide. See what it is written: the specificity of some common methods for fragmenting polypeptide chains. ये fragmenting means to cut, polypeptide chain को cut करने के लिए यूज करते हैं this proteases enzymes. तो basically sequence determination के लिए यूजफुल होता है ये, right? इससे रिलेटेड क्वेश्चंस भी बहुत आते हैं. Okay, so from here we are starting with primary structure. So these are the two important angles, basically dihedral angles. Remember this, dihedral angles. Okay, this is 5 and this is psi. Okay, 5 and psi angles. You can see these are also known as torsion angles. It is written here, torsion angles of the polypeptide backbone. So C alpha and N bond is called as 5 and C alpha and C bond known as psi. Okay, so by convention both 5 and psi are 180 degrees in the conformation shown and increase as indicated when the peptide plane is rotated in the clockwise direction as usual from C alpha. Okay, so this you have to remember. Torsion angles मतलब restricted movements जैसे होता है वैसा effect इस angles पर होता है, right? So the range, it ranges from -180 to +180. Okay, and these are very important. So as we know primary structure में sequence होता है, like this is N terminus, this is C terminus, यहाँ पे lysine है, leucine है. Okay, then glycine है, right? So basically suppose this is a 3 peptide chain, 3 amino acid का sequence है. Got it? So this is the primary structure, but the amino acids are joined by peptide bonds, as we know. So these angles, 5 and 5 angles, are important for a secondary structure as well, right? So secondary structure के लिए भी important है. So let's see now एक overview of protein structure. Some points. A typical protein usually has one or more stable three-dimensional conformation that reflects its function. Some proteins have segments that are intrinsically disordered. Okay, disordered होता है, but are nonetheless essential for function. मतलब disorder है structure में, फिर भी वो as a function it is essential, right? Whereas non-peptide covalent bonds means peptide bond नहीं है, but covalent bond है ऐसा. Particularly disulfide bond. See, disulfide bond is formed between cysteine, right? Can play a role in stabilization of some structures. Okay, stabilization में इसका role है disulfide bond का. Proteins are stabilized largely by multiple weak non-covalent interactions and forces. मतलब non-covalent भी है as well as covalent है. At covalent में peptide bond, disulfide bond and non-covalent interactions भी है. The hydrophobic effect derived from, let's see what is the hydrophobic effect. That is very important. Okay, see, derived from increase in entropy. Entropy is what? Randomness or disorderedness. तो entropy जब increase होती है तो hydrophobic effect हमें दिखता है, right? Of the surrounding water when non-polar molecules or groups are clustered together makes the major contribution. See, इसका major contribution है to stabilize the globular form of most soluble proteins. Got it? तो globular form को stabilize करने में इसका role है. Hydrophobic effect. Okay, hydrophobic effect means we know इसमें non-polar जो molecules है और groups है का clustering होगा, right, around the water. Next point, hydrogen bonds and ionic interactions are optimized in the thermodynamically most stable structures, right? तो इसका भी optimization होता है hydrogen bonds as well as ionic interactions. Next is van der Waals interaction involve attractive forces between molecular dipoles. Okay, van der Waals interaction भी बहुत important है, right? Over short distances. Individually, these interactions are weak. Okay, मतलब individual bond को अगर हमने देखा तो ये weak होता है, but as a overall structure को देखा, right? It is written. If we see it when combined in well-packed protein structures, provide significant effects and stabilization. So as a overall, it is very important. Significant effects provide करता है and stabilization भी देता है पूरे protein structure को. Okay. The nature of the covalent bonds in the polypeptide backbone places constraints on the structure. Right? The peptide bond has a partial double bond character. Partial double bond character है that keeps the entire 6-atom peptide group in a rigid planar configuration. Then the NC alpha and C alpha C जो हमने देखा, okay, can rotate. See, these angles or bonds can rotate to define the dihedral angles phi and psi respectively. Although permitted values of phi and psi are limited by steric clashes and other constraints. Okay, मतलब कुछ ही values है permitted values जिसको हम permitted values बोलते हैं of phi and psi. These are limited by steric clashes and other constraints. So this is overview of protein structure. Now we will go to secondary structures, right? सेकेंडरी स्ट्रक्चर यहाँ से स्टार्ट होगा। तो सेकेंडरी स्ट्रक्चर में फर्स्ट इज अल्फा हेलिक्स, यू कैन सी दिस, ये अल्फा हेलिक्स है। आई एम सजेस्टिंग यू टू प्लीज रीड दिस ऑल द डिटेल्स attached with the picture. Okay, I will be giving highlights in the video, but जो ये अटैच किया है मैंने वो सब आप खुद से पढ़ लेना, right, while this video or afterwards, by removing screenshot and save this because this is important. Mostly diagram से ही questions आए हैं. Okay, now of this alpha helix, to tell alpha helix, the first primary structure, इसमें secondary structure, sorry, right-handed RH is the commonly found. Okay, common. And LH जो left-handed है, that is not seen. Theoretically possible है left-handed वाला, but proteins में हमें नहीं दिखने को मिलता. Why? Because it is not stable compared to right-handed structure. Okay. Now from this is very important. The repeat unit. See, it is written here. The repeat unit is a single turn of helix with 3.6 residues. Okay, ये important है. बाकी ये पूरा रीड करना, ये सब कुछ रीड करना है. नीचे का जो इंफॉर्मेशन है this of every picture you should read. Now see, this is a helix dipole. Okay, this is a helix dipole. Basically क्या है, on the upper end we can see C terminus. Okay, negatively charged. तो यहाँ पे डेल्टा नेगेटिव चार्ज है, and on the opposite end there is a N terminus, amino terminus, जहाँ डेल्टा पॉजिटिव चार्ज है. ओके, तो this gives rise to dipole basically. and अभी यहाँ पे अमीनो एसिड्स कैसे होंगे फिर in this structure? Very logical it is that अगर यहाँ पे निगेटिव चार्ज है तो इसको बैलेंस करने के लिए, स्टेबलाइज करने के लिए यहाँ पे पॉजिटिव चार्ज होना कंपलसरी है. तो पॉजिटिवली चार्ज अमीनो एसिड्स होंगे सी टर्मिनस पर टू बैलेंस द नेगेटिव चार्ज. सेम हियर ऑन द अपोजिट साइड, यहाँ पे इन्वायरमेंट पॉजिटिवली चार्ज है तो अमीनो एसिड्स नेगेटिवली चार्ज चाहिए टू बैलेंस द कंडीशन. ओके, सो कार्बोक्सी टर्मिनस पर हमें पॉजिटिवली चार्ज अमीनो एसिड्स ज्यादा मिलेंगे. ओके, मतलब इसका मतलब ऐसा नहीं है कि नेगेटिवली चार्ज होंगे ही नहीं, पॉजिटिवली चार्ज का अमाउंट ज्यादा होगा कंपेयर टू नेगेटिवली चार्ज. सेम ईयर जो अमाइनो टर्मिनस है उस पर नेगेटिवली चार्ज अमाइनो एसिड्स ज्यादा होंगे कंपेयर टू पॉजिटिवली चार्ज. गॉट इट? नेक्स्ट स्ट्रक्चर इज बीटा. ओके, बीटा कन्फर्मेशन ऑफ पॉलीपेप्टाइड चेन. जो ए पिक्चर है दैट इज बीटा स्ट्रैंड. बीटा स्ट्रैंड इज सीन जब सिंगल चेन है. When there are multiple chains, then it gives rise to sheet. अभी anti parallel means you can see arrows, anti parallel है. Okay, and parallel में same side, right? Anti parallel में opposite, parallel में same. So this is about beta strands and beta sheets. Single chain है तो beta strand, and multiple, when there are multiple chain, then it gives rise to sheet. Here next is connections. मतलब जो बीटा शीट्स है उसको कनेक्ट करते हैं फिर लूप्स, राइट? लूप्स और टर्न्स वी कैन से, which are flexible. See this, A picture is what? Anti-parallel strands may be connected by small loop. This is anti-parallel. Anti-parallel. And this is parallel. Parallel strands require a more extensive crossover connection that almost always have a right-handed helical sense, as is shown because it better accommodates a beta sheet's inherent right-handed twist. Okay. Now these are the basically angles. See, idealized 5 and 5 angles for common secondary structures in proteins. Okay, just to show you, not to remember, just to see. See, every structure जो हमें दिखता है यहाँ पे, like alpha helix, 5 angle is 57 degree and psi angle is 47 degree. Okay, beta conformation में anti parallel 130, 135, यहाँ पे negative है, यहाँ पे positive है, यहाँ पे दोनों negative है alpha helix में. Okay, parallel में भी. Okay, same you can check. Collagen triple helix, beta turn, right? Beta turn means this will form loop for connecting connections. So now this is very important, Ramachandran plot. Okay, Ramachandran plot, यहाँ से भी बहुत questions आते हैं. This is very important for the secondary structure determination. Dependent on, okay, ये भी dependent है on those dihedral angles. You can see plot किया है -80 to +80, right, on both the axis, x-axis, y-axis. So you have to remember this. Anti-parallel beta sheet कहाँ पे आते हैं? Parallel वाला. Okay, exact points you have to remember. Then collagen triple helix. You can see, right, twisted beta sheet. यहाँ पे पूरा ये जो है is this one. Then left-handed alpha helix. Okay, right-handed alpha helix यहाँ पे है. Got it? So what it is written here? The values of phi and psi for various allowed conformations and secondary structures are shown. Peptide conformation deemed possible are those that involve little or no steric interference based on calculation using known van der Waals radii and dihedral angles. Other types of Ramachandran plot make different assumptions. The areas shaded dark blue represent conformation that involves no steric overlap, see, and are fully allowed. जो dark region है. Medium blue indicates conformational permitted if atoms are allowed to approach each other by an additional 0.1 nm, right? मतलब अगर approach किए तो allowed permitted values on a slight clash. See, the lightest blue indicate conformation that are permissible if a very modest flexibility, right, is allowed in the omega dihedral angles. That describes the peptide bond itself. So 5 and psi के साथ omega angle भी होता है. Okay, that is basically peptide bond itself. Generally constrained to 180 degree. Okay, and the white regions are conformation that are not allowed. जो white है. Although left-handed alpha helices extending over several amino acid residues are theoretically possible, they have not been observed in proteins. As I told you, जो left-handed है theoretically possible है, but proteins में हमें नहीं देखने मिलते. The asymmetry of the plot results from L stereochemistry of amino acid residues. The values of 5 and psi for all the amino acid except glycine in the enzyme pyruvate kinase isolated from rabbit are overlaid on the plot of allowed conformation. मतलब ग्लाइसिन को एक्सक्लूड किया है. Why? Because it is flexible. See, the small flexible glycine residues were excluded because they frequently fall outside the expected ranges. right? जो expected range है blue range उसके बाहर एग्जिस्ट करते हैं, तो ग्लाइसिन को एक्सक्लूड किया है, मतलब इंक्लूड नहीं किया है. ओके, सो दिस इज वेरी इंपॉर्टेंट. ये इंफॉर्मेशन जो इमेज के साथ अटैच होते हैं, दैट इज ऑल्सो वेरी इंपॉर्टेंट व्हेन यू स्टडी, राइट. सो आई सजेस्ट रीड करो. ओके, मैंने आई विल नॉट बी रीडिंग ऑल द इमेजेस, यू रीड बाय योर एंड. ये भी है वन मोर, यू कैन सी इसमें भी सेम. दिस इज एंटी पैरेलल collagen triple helix. This is left-handed alpha helix and this is right-handed alpha helix. Okay, ये मेमोराइज करना बहुत इंपॉर्टेंट है. रामचंद्रन प्लॉट. नेक्स्ट इज CD spectroscopy. This is also very important for secondary structure determination. I will read this also. These spectra show polylysine, okay, entirely as alpha helix, as beta conformation, or in an unstructured denatured state. The y-axis unit is a simplified version of the units most commonly used in CD experiments. Since the curves are different from alpha helix, beta conformation and unstructured, the CD spectrum for a given protein can provide a rough estimate for the fraction of the protein made up of the two most common secondary structures. The CD spectrum of the native protein can serve as benchmark for the folded state. Okay, जो folded state जो होता है प्रोटीन का, उसके लिए important है बहुत, useful for monitoring denaturation or conformational changes brought about by changes in solution condition. Okay, so you can see alpha helix है, right? So beta conformation and this is unstructured. This comes in the techniques part basically, but ये जो प्रोटीन से रिलेटेड जो चीजें हैं फ्रॉम अ ग्राफ और फ्रॉम अ चार्ट, ये बहुत इंपॉर्टेंट है बिकॉज ये विजुअल इंफॉर्मेशन क्वेश्चंस में पूछते हैं. ओके, सेम ऐसे ड्रॉ करेंगे and they will tell to identify and you should identify which is the which one. तो ये सबसे जो ऊपर है ये अल्फा हेलिक्स का है, नीचे भी जा रहा है देखो बहुत. ओके, एज वेल एज द नेक्स्ट वन जो बीटा कन्फर्मेशन है, ये सेकेंड है. ऐसे स्ट्रक्चर जो ब्लैक है अनस्ट्रक्चर्ड, नीचे से ऊपर, निगेटिव से ऊपर जा रहा है. Alright, so this you should remember. So let's see the summary of secondary structures. Secondary structures is the local spatial arrangement of main chain atoms in a selected segment of a polypeptide chain. It can be completely defined by the pi and psi angles of all the amino acids in that segment. Okay, ये तो हमने देखा definition, angles भी देखे. In the alpha helix, the repeating unit is a single helical turn of 5.4 angstrom or 3.6 amino acids. Important detail: the common form found in protein is right-handed with the amino acid R groups protruding away from the helical backbone. The propensity of a protein segment to form alpha helix depends on composition of its amino acids, जब कौन से amino acids है, and amino acid positions relative to one another, relative to the helical dipole जो हमने देखा. In the beta conformation, amino acids are extended in a zigzag fashion. Okay, sheet arrangement. When several beta strands are arranged adjacent to one another, they can form either parallel or anti-parallel beta sheets. Turns or loops connect segments of alpha helix or beta strands. Beta turns, which often contain glycine or proline residues, tend to connect segments of anti-parallel beta sheets. So ये important point है about glycine and proline. I will write here separately. Okay, glycine flexibility देता है, flexibility, and proline it introduces kink. Kink means bend in the structure. Okay, so these are not found in alpha helix. अल्फा हेलिक्स को ये destabilize करेंगे. Okay, so these are not found in alpha helix. This is very important point you have to remember. Destabilize करते हैं alpha helix को. So these are found in basically loops and turns. Okay, found in loops and turns. जो हमने ये point अभी पढ़ा. Okay, see here. Glycine और proline residues beta turns में present है. Okay, and this connect segments of anti-parallel beta sheets. Very important. This you should remember. The Ramachandran plot is a visual description of the combination of 5 and 5 dihedral angles that are permitted in a peptide bond. See, permitted means जो allowed values है उसका combination, and those that are not permitted उसका भी due to steric constraints. Dihedral angles that define the alpha helix and beta conformation are found only within certain regions of the plot. हमने देखा है. Circular dichroism spectroscopy is a method for assessing common secondary structure. Determination करने के लिए important है ये method. Monitoring folding in proteins based on absorption of circularly polarized UV light. Okay, so this is also absorption of basically circularly polarized UV light. Right, this principle is also very important, you should remember. So अभी on this basis हम क्या बोल सकते हैं, जो अल्फा हेलिक्स है उसका absorption high है, right, compared to beta conformation. So now we are going to tertiary structures. Tertiary structure basically represent three-dimensional folding, right, as we saw. In considering this higher levels of structures, it is useful to designate the major groups into which many proteins can be classified. Okay, so basically three-dimensional structure जो हम देखते हैं ना, so this is combination of tertiary structures and quaternary structures as well. So not only tertiary structures, quaternary structures भी इसमें आएंगे. Right, so इसमें types है fibrous protein and globular protein. So fibrous protein is what? इसमें polypeptide chains arranged in long strands or sheets. Okay, I will give you a tip here. Remember, F for fibrous, F for filamentous. Okay, मतलब filament like, chain like structure हमें दिखता है, right? And जो next type है globular protein with polypeptide chains folded into spherical और globular shape. Globular में हमें spherical और globular shape मिलता है, folded structure होता है properly. Then the next type is membrane protein. जो हमने देखा है membrane protein membrane में inserted होते हैं, right? Peripheral integral with polypeptide chain embedded in hydrophobic lipid membranes. Right? Embedded means inserted in membrane. Next type is intrinsically disordered protein with polypeptide lacking. Very important. See, intrinsically disordered protein, disordered क्यों है? Basically because इसमें stable tertiary structures नहीं होते. Okay. So now this ये जो three groups है, which three groups? Fibrous protein, globular protein and intrinsically disordered protein. Because membrane protein तो सेल बायोलॉजी में हमने डिस्कस किया है, राइट। तो दिस 3 इंपॉर्टेंट प्रोटीन जो क्लासेस है, दिस आर स्ट्रक्चरली डिस्टिंक्ट, मीन सेपरेट है, डिफरेंट है बहुत। फाइब्रस प्रोटीन यूजुअली कंसिस्ट ऑफ सिंगल टाइप ऑफ सेकेंडरी स्ट्रक्चर, ओके, एक सिंगल टाइप ऑफ सेकेंडरी स्ट्रक्चर होता है, एंड द टर्शरी स्ट्रक्चर इज रिलेटिवली सिंपल, राइट। शीट अरेंजमेंट है, लॉन्ग स्टैंड अरेंजमेंट है, सो इसका सिंपल स्ट्रक्चर है फाइब्रस प्रोटीन का। ग्लोबुलर प्रोटीन्स ऑफन कंटेन सेवरल टाइप्स, मतलब इसमें मिक्सचर्स होता है ऑफ सेकेंडरी स्ट्रक्चर। इंटरसिंगुलर डिसऑर्डर्ड प्रोटीन में लैक सेकेंडरी स्ट्रक्चर एंटायरली, ओके, सेकेंडरी स्ट्रक्चर होता ही नहीं है। द ग्रुप्स ऑल्सो डिफर फंक्शनली, द स्ट्रक्चर्स दैट प्रोवाइड सपोर्ट, shape and external protection to vertebrates are made up of fibrous protein, okay, मतलब external protection means what, जो hair है, support means bone, collagen वगैरह, that is of fibrous protein, shape provide करने वाले, support provide करने वाले structures fibrous protein से बनते हैं, and enzymes are, most enzymes are globular proteins, whereas regulatory proteins can be globular, ओके, जो रेगुलेटरी प्रोटीन्स है ग्लोबुलर हो सकते हैं, डिसऑर्डर्ड हो सकते हैं, और कंटेन बोथ ग्लोबुलर एज वेल एज डिसऑर्डर्ड segments. Got it? Now this is another important table, secondary structures and properties of some fibrous proteins. Okay, अभी स्ट्रक्चर में पहला अल्फा हेलिक्स, क्रॉस लिंक बाय डाइसल्फाइड बॉन्ड्स, tough insoluble protective structure of varying hardness and flexibility. Examples of occurrence: α-Keratin of hair, feathers and nails. Got it? Next is beta conformation, characteristics of flexible filament, right? Alpha helix tough है, hard है, beta conformation soft flexible है. Example: silk fibron, and collagen triple helix high tensile strength without stretch, okay, high tensile strength, collagen of tendon bone matrix. So just I will show you the diagrams. So these are the two chains. Okay, alpha helix keratin का स्ट्रक्चर है, इंपॉर्टेंट है, इसमें डाइमर है, these dimers come together to form protofilament, but this is a two chain coil structure, this come together to form protofilament, and then protofilaments come together to form protofibril. So ये स्ट्रक्चर बहुत इंपॉर्टेंट है, but it here लेफ्ट हैंडेड इंटरवुंडिंग है इसमें of alpha helices. So I will recommend read this, very important. Okay, next is collagen structure. तो collagen को क्या बोलते हैं? ट्रिपल हेलिक्स. ट्रिपल हेलिक्स मीन्स व्हाट? देयर आर थ्री चेन्स, थ्री अल्फा हेलिसेस है, राइट? अभी इसमें ना सीक्वेंस है इंपॉर्टेंट, हियर ग्लाइसिन XY, so where X is often proline and Y is hydroxyproline. Okay, you should remember this, right? This is most important. बाकी का रीड करना, ओके? कोलेजन फाइब्रिल्स you can see here, ये ट्रिपल हेलिकल स्ट्रक्चर सी, you can see here क्रॉस स्ट्राइएशंस as well. जो ये लाइंस दिख रही है सर कॉल्ड क्रॉस स्ट्राइएशंस, सब लिंकिंग है, क्रॉस लिंकिंग. क्रॉस लिंकिंग में लाइसिन, हिस्टिडीन एंड 5-हाइड्रोक्सीलाइसिन का रोल है. Okay, also one important information, this is again a silk, basically protein fibroin, okay, which consists of beta chain, right? Anti parallel beta sheets है, देखो, rich in alanine and glycine residues. Got it? So you have to read everything by your side. That is very important. Okay, I'm attaching all the important things in this video of protein. Now ये जो टेबल है इसमें जस्ट एक अप्रोक्सीमेशन दिया है of alpha helix and beta conformation proportion you will find in different proteins. Like there is a protein chymotrypsin enzyme है, alpha helix 14% मिलेगा and beta conformation 45%. Premier carboxypeptidase ये enzyme है एक 38% alpha helix, beta conformation 17%. Just to give an idea, this is not that important, just to show you. Okay. ये एक प्रोटीन में अलग अलग सेकेंडरी स्ट्रक्चर हमें मिलते हैं. The ratio is not equal, proportions matters. Okay, different है and that is functional significance भी है उसका. Now this is when we see the 3D structure, तो ऐसे motifs present होते हैं. जैसे motif, मोटिफ बीटा बैरल हमने देखा है, बीटा बैरल फोर फॉर्म करते हैं, राइट. बीटा बैरल, ये बीटा अल्फा बीटा लूप है, तो ये अल्फा हेलिक्स है एंड दिस इज बीटा का शीट, इसको लिंक किया है यहाँ पे लूप ने. Got it? From terminology only you can understand. Okay, this information written you should read. Now this is again a beta beta alpha loop. You can see here which is a part of this barrel structure. जो ये पूरा बैरल है इसका एक छोटू सा पार्ट है बीटा अल्फा बीटा लूप, राइट. दिस इज अल्फा बीटा बैरल एंड यू कैन सी हियर ये जो टू प्रोटीन्स सब यूनिट्स हैं. ओके, सो दिस इज अ सब यूनिट प्रोटीन, टू सब यूनिट्स जॉइंट, राइट. तो स्ट्रक्चरल डोमेन्स, इसको हम डोमेन्स बोलते हैं. टू डोमेन्स है इन द पॉलीपेप्टाइड ट्रोपोनिन सी, मतलब प्रोटीन है ट्रोपोनिन सी, इसके दो डोमेन दिखाए हैं. बेसिकली कैल्शियम बाइंडिंग, राइट. दिस इज द कैल्शियम बाइंडिंग प्रोटीन associated with muscle, has two separate calcium binding domains shown here in brown and blue. Got it? Okay, now this is very important, you have to understand. K analysis है basically of intrinsically disordered carboxyl terminus of P53 protein to its binding partners. मतलब डिसऑर्डरनेस को हम कैसे एनालाइज करते हैं बेसिकली विद एग्जांपल ऑफ P53 प्रोटीन. तो अभी ना यहाँ पे N to C टर्मिनस दिया है, N टर्मिनस ब्लू कलर, C टर्मिनस रेड कलर. तो ये जो बीच का रीजन है दिस इज ऑर्डर्ड. ओके, ये ऑर्डर्ड है एंड जो एक्सट्रीम है ना ब्लू एंड रेड दिस आर डिसऑर्डर्ड. ओके, तो ये डिसऑर्डर्ड रीजन है, राइट. इसमें यही लिखा है सब. तो दिस ग्राफ रिप्रेजेंट्स अ प्लॉट ऑफ पॉन्डर. So what is PONDR? Predictor of natural disordered regions. जो disordered regions है उसका prediction हम कर सकते हैं by using this graph. Okay. So PONDR is one of the best available algorithm for predicting the likelihood that a given amino acid residue is in a region of intrinsic disorder based on the surrounding amino acid sequence and amino acid composition. तो इसका स्कोर अगर 1 है ना, इंडिकेट प्रोबेबिलिटी ऑफ 100% दैट अ प्रोटीन विल बी डिसऑर्डर्ड. ओके, जो 1 है वो दिखाता है 100% डिसऑर्डर प्रोटीन. दिस यू शुड रिमेंबर. मतलब 0 ये ऑर्डर होगा, राइट. ऑर्डर होगा 0 to 1, इसमें वेरिएशंस हो गए, different regions of ordered and disordered. Got it? So here it is written central domain is ordered and जो amino terminal that is blue, carboxyl terminal red are disordered. Very end of carboxyl terminal region has multiple binding partners and folds when it binds to each one of them. Right? मतलब जो C terminus है इसमें 4 अलग अलग प्रोटीन बाइंड हो सकते हैं. So that is written multiple binding partners. मतलब जब के सी टर्मिनस वाला रीजन जब अलग अलग प्रोटीन्स को बाइंड होगा तो ये फोल्ड्स भी प्रोड्यूस करता है. राइट, फोल्ड्स भी प्रोड्यूस करते हैं. तो दिस इज समरी. टर्सरी स्ट्रक्चर is the complete three-dimensional structure of a polypeptide chain. Many proteins fall into one of the four general classes based on the tertiary structure, as we saw: fibrous, globular, membrane, and disordered. Insoluble fibrous proteins such as those that make up keratin, collagen, and silk have simple repeating elements of secondary structure. Right? In some fibrous proteins, the individual polypeptide chain interact to form complex quaternary structure. Okay, मतलब secondary structures का interaction होता है to form complex quaternary structure like coil for strength and flexibility. Okay, जो coil है, that structure is basically quaternary structure. Intertwining से बनता है. Same here collagen triple helix जो बनता है, that is also quaternary structure. Globular protein have more complicated tertiary structures. Fibrous से ज़्यादा complicated structure globular का है, often consisting several types of secondary structures in the same polypeptide chain and fulfill many different functional roles in the cell. The first globular protein structure to be determined by X-ray diffraction method was that of oxygen binding protein myoglobin. तो ये first determine किया था, very important, you should remember. The myoglobin structure revealed for the first time how protein structure and functions are connected. The complex structure of globular protein can be analyzed by examining of folding patterns called motifs or folds. Okay, इसको एनालाइज करके हम ग्लोबुलर प्रोटीन्स को एनालाइज कर सकते हैं. The many thousands of known protein structures are generally assembled from a repertoire of only a few hundred motifs. Domains are regions of polypeptide chain that can fold stably and independently. Right? Domains independent units है, join होते हैं, assemble होते हैं. Some proteins or protein segments are intrinsically disordered. Okay, intrinsically disordered means what? Lacking three-dimensional structures. Okay, why it is called disordered? मतलब हम टिप याद रखने के लिए रख सकते हैं. Disordered है basically because it is not having three-dimensional structure, is lacking. Okay, these protein often have distinctive amino acid composition that allow a more flexible structure. Flexible structure है which is critical for their biological function. Based on structural similarities, protein can be organized into families and superfamilies. Right, families and superfamilies में हम organize कर सकते हैं proteins को based on structural similarities, which are informative about protein function and evolution. So this is important for function and evolution. What? Organization of proteins. Quaternary structure results from interaction between subunits of multi-subunit, that is multimeric protein, or large supramolecular assemblies. Some multimeric proteins are composed of repeated subunits. Okay, repeated subunits है तो क्या बोलते हैं? प्रोटोमर्स. राइट, एंड अलग अलग है तो मल्टीमर्स. ओके, मल्टी subunit complex. Right. So now we will see how the tertiary structures are determined. So 3D structure determination के लिए first is X-ray crystallography. Okay, X-ray crystallography very important. What it does basically, X-ray diffraction pattern are generated from a crystal of a protein, and this data extracted from this diffraction patterns are used to calculate a three-dimensional electron density map. Okay, electron density map produce करते हैं. Next is NMR spectroscopy. NMR spectra, right? NMR spectra 2D डायमेंशनल डेटा से 3 डायमेंशनल डेटा जनरेट करता है. Okay, 2 dimensional data used to generate a 3 dimensional structure. So here globin is shown as an example. Next is cryo EM. Cryo electron microscopy. EM is electron microscopy. This is also very important for analysis of 3 dimensional structure. So here protein shown is GROEL. Folding के लिए important है. GROEL. GROW stands for growth. Okay. And this is in the bacterial cases. Bacterial protein. Next is funnel. Energy funnel. The funnel diagram बोलते हैं इसको. And this is for protein folding, right? Protein folding. अभी ये A पिक्चर में सिंपल डायग्राम दिख रहा है हमें. तो this stands for no intermediates. ओके, इंटरमीडिएट नहीं है यहाँ पे. एंड यहाँ पे जो B पिक्चर है इसमें बहुत इंटरमीडिएट्स है प्रोटीन फोल्डिंग के प्रोसेस में. बट इट सी जो B पिक्चर है मल्टीपल फोल्डिंग इंटरमीडिएट्स एग्जिस्ट, right? And there are multiple pathways also, right? So here we see the dynamics, thermodynamics basically. Delta G represented by depth of the funnel. जो N है डेल्टा G रिप्रेजेंट करता है नीचे. N stands for native structure. Native structure N. At the bottom where there is lowest free energy point. Got it? So protein denaturation and folding we will be studying in cell biology section in detail. यहाँ पे हम एक overview लेंगे. Again, the maintenance of steady state collection of active cellular protein required under a particular set of condition. मतलब जो continuous maintenance जो चलता है प्रोटीन का from synthesis to degradation, that is called as proteostasis, right? So this involves elaborate set of pathways and processes that fold, refold and degrade polypeptide chain. The three-dimensional structure and function of most protein can be destroyed by denaturation. Right? Denaturation से destroy कर सकते हैं प्रोटीन का three-dimensional structure and study कर सकते हैं basically about the function of the protein, demonstrating a relationship between structure and function as well as heat, extremes of pH, right? Organic solvents, solutes and detergents can all be used to denature proteins. Some denatured proteins can renature spontaneously. मतलब कुछ प्रोटीन्स डीनेचर करने के बाद रीनेचर फिर से हो सकते हैं. मतलब they can regenerate themselves from denaturation to renaturation to form biologically active proteins again, showing that tertiary structure is determined by amino acid sequence. Right now, tertiary structure is also determined by amino acid sequence, the composition, what amino acids are there. Protein folding occurs too fast for it to be completely random process. Instead, protein folding is generally hierarchical. Initially, regions of secondary structure may form. Right? Initially, मतलब starting में secondary structures बनते हैं, followed by folding into motifs and domains. Large assembles of folding intermediates are rapidly brought to single native conformations. Got it? अभी for many proteins, folding is facilitated by HSP70 chaperones. राइट? प्रोटीन फोल्डिंग में रोल है इसका. हमने देखा है प्रोटीन जब ट्रांसपोर्ट होता है तब अनफोल्डेड स्टेट पे रखने के लिए भी इम्पोर्टेंट है HSP, एज वेल एज टू फोल्ड दैट प्रोटीन आफ्टर प्रॉपर ट्रांसपोर्ट इनटू दैट स्पेसिफिक ऑर्गेनेल. राइट? मतलब ट्रांसपोर्ट होने के बाद प्रोटीन प्रॉपरली फोल्ड होता है बाय दिस HSP प्रोटीन्स. HSP70 का मेजर रोल है एज वेल एज बाय chaperones. Disulfide bond formation and cis-trans isomerase of the propeptide bonds can also be catalyzed by specific enzymes during folding. Okay, protein disulfide isomerase होता है, prolyl cis trans isomerase. Protein misfolding is the molecular basis for many human diseases. Right, मतलब प्रोटीन अगर प्रॉपरली फोल्ड नहीं हुआ, मिसफोल्डिंग की वजह से डिजीजेस भी हो सकते हैं. So these are cystic fibrosis, amyloidosis. Amyloidosis means what? जिसमें अमाइलॉइड का एक्यूमुलेशन होता है. तो अमाइलोइडोसिस में अल्जाइमर डिजीज है, पार्किंसंस है. So let's see. This is again a very important chart. Right? I will mark here very important. And इसमें क्या दिया है? Some protein misfolding diseases दिए हैं. तो अल्जाइमर डिजीज वेरी इंपॉर्टेंट है, जिसमें डिफेक्टिव प्रोटीन अमाइलॉइड बीटा प्रोटीन है. राइट, देन ALS, अमायोट्रोपिक लैटरल स्क्लेरोसिस, जिसमें प्रोटीन सुपर ऑक्साइड डिसम्यूटेज इज डिफेक्टिव. हंटिंगटन डिसीज एक इंपॉर्टेंट है बहुत. हंटिंगटन डिसीज वेयर द डिफेक्टिव प्रोटीन इज हंटिंगटिन विथ पॉलीग्लूटामेट एक्सपांशन. राइट, मतलब पॉलीग्लूटामेट, ग्लूटामेट जो है उसका रिपीट्स बहुत इंक्रीज होता है, नंबर ऑफ रिपीट्स इंक्रीजेस. Then next is Parkinson disease. Protein involved alpha-synuclein. So now again this is about determination. First, X-ray crystallography. Protein molecules are crystallized in well-ordered orientation that diffract X-rays. The patterns and intensities of the refracted X-rays depend on the structure of the protein and its crystalline properties. Mathematical methods can then reconstruct the protein structure that produces a particular diffracted pattern. So X-ray diffraction very important for 3D structure analysis, right? हमने देखा इलेक्ट्रॉन डेंसिटी मैप प्रोड्यूस करता है. Then NMR is what? Nuclear magnetic resonance. Is often carried out molecules in solution and yield information about atomic nuclei. See, in short, to tell, NMR gives information about chemistry of the protein. Okay, so इसमें atomic nuclei है and the chemical environment. तो इसमें क्या क्या है? Protein structures can be computed from NMR data. NMR data से compute मतलब डेटा हम आइसोलेट कर सकते हैं using hundreds of distance and geometric constraints obtained from multi-dimensional NMR experiments. Okay, multi-dimensional means what? One dimensional, two dimensional analysis करके then एक हम 3-dimensional analysis, okay, 3D structure data determine कर सकते हैं using the 2D data. Got it? Next is biomolecules are frozen in vitreous ice for imaging by cryo electron microscopy. Cryo means what? See, cryo stands for cold. You should remember this. तो कोल्ड इन्वायरमेंट यहाँ पे इम्पोर्टेंट है. तो जो बायोमोलिक्यूल्स है जैसे कि प्रोटीन को फ्रोजन आइस में रखना इम्पोर्टेंट है. The individual molecules are then identified and computationally sorted. The sorted two-dimensional images, see यहाँ पे sorted two-dimensional images ही है, right? 2D data ही है that are combined using again computer to produce a 3-dimensional structure. मतलब 2D images से 3-dimensional structure produce कर सकते हैं. तो NMR में भी 2D data है, इसमें भी 2D data है, but it can produce 3D data. Got it? So now as a overall, this is the levels of protein structure. You can see here primary structure में just sequence होता है अमीनो एसिड का. सेकेंडरी स्ट्रक्चर में अमीनो एसिड का what we can say arrangement we can see, and tertiary structure में proper folding in the 3D space, right? See here, like this is alpha helix getting associated here in the tertiary structure polypeptide chain. So this is a part of this tertiary structure, right? 3D structure, and again ये एक sub unit है, right? This overall is a one sub unit of this quaternary structure where you can see assembled sub unit. Assembled मतलब join हुआ है as a overall to function as a unit. Got it? तो प्राइमरी स्ट्रक्चर में क्या होता है? Sequence of amino acids linked together by peptide bond, and it includes any disulfide bonds as well. Resulting polypeptide can be arranged into units of secondary structure such as alpha helix. The helix is a part of tertiary structure of the folded polypeptide, which is itself one of the subunits that make up the quaternary structure of the multi-subunit protein. In the case of hemoglobin, you can see there are 4 subunits here joined together, right? Next, table important hai again. I will mark here conjugated protein. Conjugated means what? मतलब इसमें non amino acid part भी होता है, right? That is what is called conjugated. So like class lipoprotein. लिपोप्रोटीन में हम लिपोप्रोटीन में लिपिड होता है along with amino acid, lipids are present. So basically this other group present is called as prostatic group, right? Prostatic group बोलते हैं. So we can see these are the examples. In the next glycoprotein, glyco में carbohydrates, prostatic group. Phosphoprotein में phosphate group, prostatic group है. Hemoprotein में heme होता है, that is iron porphyrin, prostatic group. Flavoprotein में flavin nucleotides. Then metalloprotein में metals है, metals. So these metals basically these are most important from this table. Okay, यहाँ से क्वेश्चंस बहुत आए हैं. आयन के केस में फेरेटिन है, जिंक के केस में अल्कोहल डिहाइड्रोजनेज ADH, कैल्शियम कैलमोडुलिन, मोलिब्डेनम डाइनाइट्रोजनेज, कॉपर कॉम्प्लेक्स 4. गॉट इट? बाकी ये सारे एग्जांपल्स भी देख लेना. लास्ट स्लाइड, प्रोटीन प्यूरिफिकेशन प्रोसीजर्स. नाउ जब भी प्रोटीन का डिटर्मिनेशन करना है, एनालिसिस करना है प्रॉपर, तो प्रोटीन को प्यूरिफाई करना वेरी इंपॉर्टेंट होता है, राइट? सो हाउ द प्रोटीन प्यूरिफिकेशन इज डन, दिस आर द सम मेथड्स, एंड दिस इज आल्सो वेरी इंपॉर्टेंट स्लाइड. यहाँ से भी क्वेश्चंस बहुत आए हैं, राइट? सो सॉल्युबिलिटी में सॉर्टिंग आउट प्रोसीजर है. सॉल्युबिलिटी में आयोनिक चार्ज में आयन एक्सचेंज क्रोमेटोग्राफी, इलेक्ट्रोफोरेसिस एंड isoelectric focusing. Ionic charge. Polarity. On the basis of polarity, the procedure used is hydrophobic interaction chromatography. On the basis of size, gel filtration chromatography and SDS page. On the binding specificity, affinity chromatography is used. Okay, so logical है, याद रखना भी, because if I tell you solubility means you can remember salt, right? Salt soluble in water. So this is how you can remember this. In the case of ionic charge, you can remember ion exchange. Electrophoresis तो है ही चार्ज के बेसिस पर, as well as isoelectric focusing भी चार्ज से रिलेटेड है. Polarity we know, non-polar is called hydrophobic, right? And polar is hydrophilic. Okay, तो इससे कुछ रिलेशन होगा. So this is hydrophobic interaction chromatography on the basis of polarity. And size, again size, gel filtration. So filtration is related again to size, as well as SDS page भी size से separation करता है. Binding, So binding is basically affinity, na? Tight binding shows the affinity. So that is how you can remember this. Okay. So that's it for this video. I hope this video helps you. If you find it helpful, do let us know in the comment section. Like this video, share this video with your friends, subscribe to our channel, and stay tuned for the upcoming videos. Thank you.